Hello AP Biology students, we're going to go over the third page of my statistics assignment. And in this page, we're going to start something called standard error bars, or standard error of the mean. On this page, we go over how to calculate and how to draw standard error bars on a bar graph. And on this page, there's only one question. It says the stomata are openings on the underside of leaves that allow for gas exchange to occur in plants. Two groups in an AP Biology class took data on the number of stomata for a sunflower plant. Group A took 6 points of data, while Group B took 16 points of data. Okay, so just let's review this. These are stomata. They're the openings on underside of leaves. We had two groups, and they took two amounts of data. What the question's going to ask is it's going to ask you to calculate the mean, or the average, standard deviation, standard error for each group. Then the question's going to ask you to graph each group's data's average as a bar graph, as well as plus and minus 2 standard error of the means. If you take a look at our table here, you can see Group A has 6 points of data, while Group B has 16 points of data. And again, it's going to ask you to calculate the mean or average, the standard deviation, and the standard error. For the mean, you add up all of the data points and divide that number by the number of data points. Now, since we've already done some calculations with standard deviation, I like to tell students that there are calculators online that will actually do this math for you, but it makes this process just a little bit easier knowing these two values. Now what we have to do is calculate standard error of the mean. You can see above here I have our equation. It's just standard deviation divided by the square root of the number of samples. So again, we're going to add in 7.6, which is our calculated standard deviation, and divide it by the square root of 6. Square root of 6 is 2.449, and that means 7.6 divided by 2.449 is 3.1. So this is our standard error of the mean. So I calculated the mean is 86, our standard deviation, remember, is 7.6, and our standard error of the mean is 3.1. Now we have to do the same thing to the other set of data. We first worked on this set of data. This set of data is going to have a different mean, a different standard deviation, and a different standard error. One of the mistakes that a lot of students make at this point is they think this standard error is going to be for everything. No, this standard error is only for this set of data, Group A's set of data. Now we have to calculate standard error for Group B's data. So again, here's our equation. We plug in 10.5, which is just what we calculated for standard deviation, divided by 16, which is the number of data points. Square root of 16 is 4. 10.5 divided by 4 is 2.6. So 2.6 is the standard error for Group B's data. So again, here's our mean, which is 88, standard deviation is 10.5, and our standard error of the mean is 2.6. So now we're going to use these two values with these two values. We actually don't really use standard deviation from this point on because we're not going to be graphing it. So this is the graph I made for the data. It's not perfect, but it does a good job of representing what we're trying to learn here. You can see Group A and Group B's data. Their averages are 86 and 88. And yes, we kind of made this graphing complete by starting at 74, but it helps us visualize these error bars a little easier. So we have Group A's data, which is again 86 is the average. We are going to use plus or minus 2 of these standard errors. So what we're going to do is we're going to make a bar-headed line plus 6.2 and negative 6.2. This is because when we graph standard error, we typically graph 2 of them, not just 1. You can see the bar-headed line is just above 92 and below 80. Group B's data has an average of 88 and a standard error of 2.6. That means we're going to go up 5.2 and down 5.2. You can see we're just above 93 and just below 83 here. The question is going to ask, is there a significant difference between these two groups of data? Explain your answer. And the answer to this question is no, there isn't significant difference between these two error bars or these two groups of data. And I like to remind anyone that's watching this that this is just for an AP Biology class. This is just intro to stats. There is a lot more that goes behind these statistical analyses of data. Basically, since these error bars overlap, they're not going to be significant from each other. I like to tell students that this error bar kind of tells us where the average could be at any point. So on one day it could be all the way up here, and another day it could be all the way down here. The same goes for this error bar and this data. The average could be anywhere between these two lines. We don't know when we do this test again where it's going to be. It could be all the way down here, all the way up here. But since there's so much variation between these two data, they're not going to be significant from each other. And this isn't like a bad thing. It just tells us what the data is actually representing.